Hello, everyone. Okay, this is Chapter 12, Section 2, RNA replication. Okay, so you should be taking notes on this. The note starts on page 17, and you guys can also use page 20 to take notes on this. So 17 I'm sorry, 17 and 19. I apologize for that. Pages 17 and 19, you're going to take notes on this, and this is the title that you're going to write at the top of the page. And make sure you note the date in which you take them. So what does DNA stand for? It stands for deoxyribonucleic acid. So this should be a review. We've been doing this in class quite a while. So this is kind of a little review thing. The number of chromosomes in every organism varies. You can see in this slide, you can see humans have 46 chromosomes. Giant sequoia trees, and if you've ever been there, they're huge trees. I mean, they're just so amazing, and they've been alive for so long. But they only have 22 chromosomes. Potatoes have 36 chromosomes. Fruit flies, these little tiny things, have 8 chromosomes. And here on the side chart, you see pea plants, 14, sunflowers, 34, cats have 38, but pufferfish 42, humans have 46 once again, and dogs have 78. So dogs have more chromosomes than humans. So what we've realized now is that chromosome number doesn't have to do with complexity or order of any sort as far as leaders on top of the food chain. So, right now, how do chromosomes compare? How does DNA compare to chromosomes? Sorry. So they're all the same genetic material, except they're in different forms and shapes. So we've got uh, basically DNA gets condensed into this stuff called chromatin. Chromatin is basically just coiled up DNA, and that little string right there, that's supposed to be DNA, a string of DNA. And DNA gets supercoiled into these things called chromosomes, and sister chromatids wind up attached through a little thing that holds them together. So these are called sister chromatids, and they're basically copies of the same genetic material, so the same DNA being copied. And chromatids are basically the single pieces, so there is different names for chromosomes depending on whether they are alone. They're chromatids, and when they're together, they're chromosomes. So this is question one. Please make sure you answer this. Or I'm gonna put this video should be on Canon, so make sure you write the answer to this question. Which is not nucleotide base in DNA? You should know this from the work that we did in class. Okay, your second question, the DNA backbone is made out of, you should know this, we did it in class. What is the base pairing rule? We did some practice ones of these, so you should know which of these is how DNA pairs up. But think of the big ladder that we did in class. Which ones went together? So how many carbons does the sugar molecule in the DNA have? We covered this in class. We numbered it. I hope you remember. Okay, until this point, we've gone over review material that we've already covered in class. Now we're going to go over how DNA copies itself, otherwise known as DNA replication. So how we make more of it. So the word to replicate, replicants or anything like that, there it basically means copies. Remember that every cell in your body needs a copy of the DNA. When you get a cut, you need to make more cells to cover that cut. So you need to make more cells. Same thing when you break a bone, you need more bone cells, so you need to copy that DNA. So every time that you do that, the cell undergoes a process called mitosis. DNA needs to get copied for the new cell. So the DNA copies itself, and hopefully you were looking down here at this little graphic. Parent cells split into two daughter cells, and this is known as mitosis. So mitosis is what we call it when cells copy themselves. What is really funny is that, like, my oldest, when he was little, I tried to teach him about mitosis, and I told him, oh, because he had a cut on his toe, and I told him, oh yeah, your cells are going over doing mitosis, and he started wiggling his toes, going, oh yes, mitosis. So I thought that was pretty funny. But anyway, mitosis is a way of copying cells. Okay, so DNA replication. What happens during DNA replication is that you get this enzyme called the DNA gets broken up. It gets basically split up, and it gets when you split up the DNA, this enzyme called DNA polymerase uh, comes around and basically adds a new base pairs to the DNA that's open. So basically, you split it up and it winds up with new partners. It's like when couples get split up and they wind up with new spouses each. Well, the DNA gets split up and each section gets a copy, a new copy being made of the other side by DNA polymerase. So DNA polymerase will lay in complementary strands. For example, here the DNA polymerase will start at the top one, 5 prime to 3 prime. So the 3 prime end starts off with a G and then an A and then a T and then an A and then a C. DNA polymerase starts putting in these nucleotides to pair up with the one that's being exposed. The same thing on the parent old strand. The other parent strand, it starts off at the 5, and DNA polymerase starts coming in and putting in C, T, A, T, G, A, and so on as the other protein DNA helicase unwinds it. So DNA helicase is the one that actually unwinds the helix. That's why it's called helicase. That enzyme is in charge of unwinding DNA, so it's DNA helicase. Okay, so hopefully you'll understand this. Okay, so kind of a really quick other review. So DNA helicase unzips DNA, the double helix. That's called helicase, double helix, kind of, that's why it's helicase. And it's an enzyme that unwinds the DNA because DNA is all coiled up in a helix, so it unwinds the helix. Then the DNA polymerase adds complementary strands, base pairs to in the 5 prime to 3 prime direction. So you should know that one strand is being created continuously. The other one is in chunks because it's opening, unzipping the right way. And the only way DNA polymerase can add new pieces is from the 5 prime to 3 prime. So one of them gets put up into pieces, but don't worry about that as long as you understand that DNA polymerase is the one that's adding the new pieces to the new strand. When this molecule of DNA is all finished, you're going to wind up with basically two identical pieces of DNA. Okay, so both DNA are identical. They're made up of the same original strand and one new strand, and the DNA polymerase made. This is called semi-conservative because you're conserving part of the old strand and you're pairing that up with a new strand. So you get part old, part new. You get that's called semi-conservative because you kind of conserve an old one, but you add a new one to it. But that makes sure that the copy is identical. So the two new strands are not completely new copies. They're half old, but because you have half old ones, you make sure that the new sides are completely perfect. Okay, I hope you were able to write down these, and I hope you got a good grade on this Canon video. And please make sure you finish all your worksheets, and I hope to see you guys soon. Bye.